Thank you very much, Professor Kudo, for a nice introduction, and congratulations on such a great success of this symposium. And first of all, I have to apologize. I have changed my title of my talk from the in glymphatic system to in neurological disorders for a couple of reasons. I think all of you are very well known, the aquaporin. I'm very pleased that you are talking about aquaporin many times since I've been working on these proteins for almost 30 years. Today, aquaporin in mammalian, we have 13 isoforms from aquaporin 0 to aquaporin 12. And today I'm going to focus on the aquaporin 4. Aquaporin 4 is permeated by water, but some of the aquaporin, such as aquaporin 3, is permeated by glycerol or urea or other small molecules in addition to water. Aquaporin 4 is distributed in the brain and especially in the astroglial cells and eye retina and kidney collecting duct and muscle. And aquaporin 4 has the quite unique features among the aquaporins. First, they make the orthogonal array like this. Aquaporin, at least there are two isoforms we call M1 and M23. M23 isoform, a little bit shorter isoform, can make this kind of array formations. And aquaporin 4 also has some cell adhesion function, homophilic interaction. And also aquaporin 4 is located in the dystrophin complex, such as syntrophin or beta-dystroglycan, alpha-dystroglycans. And as I told you, aquaporin 4 is mainly expressed in the astrocytes, but not diffusely expressed in the astrocyte plasma membrane, but exclusively expressed in the process surrounding the brain capillary blood vessel and lining the ventricles and also the surface just beneath the pia mater. This unique distribution of aquaporin 4 clearly demonstrates aquaporin 4 only expressed in the interface between blood and brain parenchyma and CSF and brain parenchyma. These distributions suggesting the role of aquaporin 4 in the exchanging the water between these compartments. And aquaporin 4 also involved in the brain edema formation in several occasions, such as stroke. And recently, aquaporin 4 has been very focused because NMO, neuromyelitis optica, this is the autoimmune disease. The patient, they have the autoimmune antibody against aquaporin 4. So today, I'm going to focus on the aquaporin 4 in neurological disorders. First, I'm going to talk about NMO spectrum disorders, and second, related to the Alzheimer's disease, and third, ALS. And neuromyelitis optica spectrum disorders initially described as a Devic diseases by using Devic in the French neurologist almost 100 years ago. And this initially we thought this is the subtype of multiple sclerosis. But in 2006, revised diagnostic criteria has been shown. And define MNO, they have the optic neuritis and acute myelitis, and at least two of these three supportive criteria. One of them is the NMO IgG. This is the autoimmune antibody recognized aquaporin 4 extracellular domain. Dr. Lennon in Mayo Clinic in the United States, she found this NMO IgG autoimmune antibody recognized aquaporin 4. So to develop the good animal model for NMO, we initially developed monoclonal antibody recognizing aquaporin 4 extracellular domain by using baculovirus display method, collaborating with Professors Hamakubo at the University of Tokyo. In using this method, we can efficiently raise the monoclonal antibody against extracellular domain of the protein, membrane protein. And we use the human or mouse aquaporin 4, either M1 longer isoform or M23 shorter isoforms, and successfully made many kinds of monoclonal antibody. For example, one of them we call E5415A recognize both M23 and M1 isoforms. And we injected this monoclonal antibody into a rat EAE model. On the left panel shows when we used NMO IgG from the patient, even if we use 40 milligram of the IgG, we can worsen the EAE clinical scores a little bit. On the other hand, if we use our monoclonal antibody, 0.1 milligram, 0.01, 0.1, and 1 milligram, those dependently worsen the clinical score of EAE. And next, we examine the pathological changes. Here is the immunohistochemistry of aquaporin 4. The middle panel shows when we use the NMO IgG from the patient, 2 milligram, 20 milligram, and 40 milligram. After injection of 40 milligram, we can see slight defect of aquaporin 4 staining. On the other hand, when we use our monoclonal antibody, 0.01 milligram, 0.1 milligram, 1 milligram, we can observe the huge lesion, aquaporin loss lesion. And we further show using the other astrocyte markers such as EAT2, glutamate transporter, and GFAP. And this aquaporin 4 loss lesion is almost identical to the lesion of EAT2 or GFAP loss lesion. This suggesting the evidence of this is the astrocyte patch. This is actually the pathological changes in the NMO patient as well. And we further examine pathological changes, especially the initial events, how NMO IgG can recognize aquaporin 4 and what happens for the next by using the high-speed AFM, atomic force microscopy. By using this, we can monitor the single molecule image. Here you can see the either monoclonal antibody NMO IgG, Y-shaped IgG. And here you can see the interaction between single IgG and aquaporin 4. As I already told you, aquaporin 4 make this kind of array formation. We overexpress aquaporin 4 and purify it and reconstitute in the black membrane, the lipid bilayer. And this is the single monomer. And in case of aquaporin 4, single monomer makes a higher array, orthogonal array formations. And if you look at here, IgG, one arm can recognize and bind to the one tetramer, and the other arm can bind and recognize and bind to the adjacent tetramer. And we did, you can see the movie, and we found this antibody can walk around on the huge array surface of aquaporin 4. And if we increase the concentration of the antibody, they can make the complex. Collision occurs and they make the complex. Once they, excuse me. Once they make the complex, they don't move around. And the IgG complex can be recognized by the complements much easier. So this is the initial event we believe of the, CDC complement dependent cell death. And also we performed characterize this antibody. So we incubate the labeled aquaporin 4 monoclonal antibody for overnight, and if we wash out, this antibody binding is gradually decreased. But if we add 10-fold molar excess of cold antibody, we can get rid of the pre-incubated labeled antibody immediately. Based on this observation, we now develop the monoclonal antibody, which has much higher affinity to aquaporin 4 compared to NMO-IgG. So we can get rid of NMO-IgG. We also have to introduce the mutation in the complement binding site of this therapeutic antibody. And now we are working on this. Next, I move on to the Alzheimer's diseases. The unique character of Alzheimer's diseases, as you know, one is senile plaques, and the other one is neurofibrillary tangles. And excuse me. And plaques, amyloid beta is accumulated. And neurofibrillary tangles, tau, abnormally phosphorylated tau accumulation is one of the trigger of these changes. So then Glymphatic system has been demonstrated 10 years ago. And in this system, aquaporin 4 plays very important role. And also sleep can regulate this glymphatic flow. The Nedergaard, they showed, their group shows aquaporin 4 is involved in this glymphatic flow, but there is still no direct evidence. They monitor the fluorescence dye or other things, A-beta, anyway, solutes. But we have to visualize the solvent water flow. At the molecular level, for example, aquaporin is only permeated by water molecules. Aquaporin 4 is not permeated by amyloid beta or fluorescence dye. So at the molecular level, they use a different pathway. But as a system, actually, aquaporin 4 may play very important role to get rid of brain waste. At the last of my talk, I will bring this up once again. And Nedergaard group, they injected radioisotope labeled amyloid beta 140 into the brain parenchyma and observed the clearance, by comparison between wild type and aquaporin 4 knockout. As you see here, in aquaporin 4 knockout, the amyloid beta clearance is delayed. Based on this, we hypothesize if we combine Alzheimer's disease transgenic mouse model and aquaporin 4 neural mice, we can see more severe phenotype. As an Alzheimer's disease model, we used several different models, but today I'm going to talk about 5FAD, which has the more A-beta 42 accumulation than A-beta 40, so they can make the huge plaque, lots of plaque. And here we observed their behavior 24 hours for 6 months. And this shows, y-axis shows their movement. And x-axis shows the circadian 24 hours. So from 6 a.m. to 6 p.m. and 6 p.m. to 6 a.m. So during the daytime, since they are sleeping, they don't move. During the nighttime, they move around, and there are three kinds of mice here. The green line indicates the Alzheimer transgenic model, and blue indicates the aquaporin 4 null, and red is the combined transgenic. And at the age of 20 weeks, there is no significant difference. And 4 weeks later, 24 weeks, you can see the progressive decrease of the behavior in this combined model. And 28 weeks and 32 weeks, progressively, this combined mice cannot move so much. And further 4 weeks later, 36 weeks, I can show you the movie. Here, there are 4 mice in one cage, and wild type, aquaporin null, and Alzheimer transgenic, and combined. And you can see one mouse is very different from the other 3. This mouse never moved. This is actually the combined model at the age of 36. And this mouse is just stay on the same position, but always reveals the seizure conversion like this. This is one of the features of Alzheimer's disease patient. And we confirm this abnormal spike in the EEG. Then we examined pathological changes in this Alzheimer model and aquaporin null combined mice. And as you see here, upper panel shows this is the just Alzheimer disease transgenic model, and the lower one in aquaporin 4 null background. You don't see any significant difference here. And actually, we quantify, and there is no significant difference. Amyloid plaque itself increased between 24 weeks and 32 weeks, but there is no difference between Alzheimer's disease transgenic and combined, red and blue. And this is the A-beta 40 as well. So insoluble A-beta 40 or A-beta 42, there's no difference. This is a little bit surprise for us. And next, we examine soluble type of A-beta 40 and A-beta 42. As you see here, after 32 weeks, we can see the difference between the 5-FAD and combined model and 40. And next, we also examined soluble A-beta 42 and 40 in interstitial fluid by using microdialysis. And left panel shows at the age of 25 weeks, and right shows the age of 33 weeks. Age 25, A-beta 42 and A-beta 40 in soluble form in interstitial fluid, there is no big difference. But at the age of 33 weeks, we can see more accumulation of either A-beta 42 or A-beta 40 soluble type in interstitial fluid in combined model compared to the 5-FAD itself. Next, we further examined using the light sheet microscopy, and left panel shows with 5-FAD alone, and right panel shows the combined, and upper panel the age 20 and lower age 36. Age 20, there is no significant difference. Maybe this blue, this is aminoglow, so showing the plaque, A-beta plaque, a little bit more A-beta plaque, but not so significant. But if you compare these two at the age of 36 33 weeks, you can see more accumulation of amyloid beta, especially lining the vessels. So in addition to the huge, larger plaque and also like CAA pathology we observed. Next, by collaborating with the Professor Iwatsubo's group at the University of Tokyo, we examined the effect of aquaporin 4 on the tau clearance. So we injected fluorescence level tau into brain parenchyma and observed clearance. And if you look at this lower left panel, you can see the transient increase and decrease of injected tau in wild type model. But in case of aquaporin 4 knockout, there's gradually slow increase of aquaporin 4 in parenchyma. And this graph shows the tau in CSF. In case of wild type, you can see transient increase of tau in CSF when we injected this tau into brain parenchyma. But aquaporin 4 knockout, you can see the gradually increased tau in CSF, suggesting tau clearance is delayed in aquaporin 4 knockout mice. Next, we injected fluorescent level tau into CSF directly through cisterna magna and monitor tau accumulation in deep cervical lymph nodes 1 hour after injection. And if you look at here, tau already moved to the lymphatic node in case of wild type 1 hour after injection. However, aquaporin 4 knockout tau accumulation in DCLN is very, very low compared to the wild type. Next, we use the PS19 transgenic model. This PS19 transgenic model, they have the abnormal phosphorylation of tau. So we again combine this with aquaporin 4 knockout mice. And here, this is the PS19 itself at the age of 9 months old. And this is the PS19 and aquaporin 4 hetero. And here, PS19 and aquaporin 4 null mice, as you see here, if you compare the PS19 and PS19 in aquaporin 4 null background, you can see the huge difference. So hippocampus and cortex, they have the huge atrophy, and we actually observed the significant loss of the neuron at the very early stages. So indicating aquaporin 4 also play important role for the tau transport. And finally, I will briefly introduce the role of aquaporin 4 in ALS mouse model. As Komaki sensei already presented, we also used SOD1 transgenic mouse model for ALS. And here's the immunofluorescence of aquaporin 4, GFAP, and merged. And this is the wild type at the age of 20 days. 200 days. And here, this is the SOD1 ALS model, age 60 days, 100 days, 120, and 140, and 170. Up to 60, we can see the typical pattern of aquaporin 4 staining, just like staining the brain vessel. But at the age of 100 day, this is not so clear, and the age 120 days, we don't see any clear staining of aquaporin 4 at the brain vessel. This is not brain vessel itself, but surrounding the astrocyte process. If we check the total protein expression level are not altered. So distribution itself is changed. And I'd like to emphasize that at the time of 100 days, GFAP staining does not change a lot. The GFAP staining pattern is changed after 140 days. So before gliosis, aquaporin 4 distribution is altered. Based on these findings, we again combine ALS dystransgenic model and aquaporin knockout. And the red line shows the this is the disease onset and age. So this combined mouse model has earlier onset of ALS. Total survival not so significantly different, but there is some statistically difference. So again, aquaporin 4 may play some role in the pathology of ALS. We are now working on the molecular mechanism of this by collaborating with Professor Misawa, the faculty of pharmacy at Keio University, School of Medicine, Keio University. And finally, we've been working on visualize the water molecule itself and, at the microscope level. So time resolution, millisecond, and spatial resolution is 1 micrometer. You know, water is too small to label the fluorescence dye. So what we did, we used Raman scattering, applied Raman scattering. Obata sensei already presented coherent anti-Stokes Raman scattering curves. When we use this microscope, we can increase the signal of Raman. And as you may know, Raman peak is very different between heavy water and water. So we can clearly distinguish water from the heavy water. So combined with rapid diffusion system, we visualized water or heavy water diffusion at the single cell level. I don't show the data today. And we also developed further. Recently, we switched to CARS to SRS. SRS is the stimulated Raman scattering, and which has a better quantitative analysis. By collaborating with Professor Ozeki at the University of Tokyo, recently we set up this microscope. The advantage of this using this microscope, we can visualize water and some fluorescence molecule, because this is the 2 photon. We can use, if we carefully choose the wavelengths, we can this SRS microscope can be used as a 2 photon excitation. So we can visualize water dynamics and some solutes molecule simultaneously. We now apply, we have recently published this data using the brain slice tissue, but now we are moving of this system to the in vivo mouse brain imaging. Very recently we were succeeded to visualize fluorescence dye and water by using this SRS method in living mouse brain. Unfortunately, I don't have the data to show this evening, but in the near future, I hope I can show you the, this simultaneous mouse in vivo brain imaging. Then we can understand what's going on in the brain water dynamics, and we can understand better of the glymphatic system. Thank you for your attention.